My name is Shaun Jackman, and I'm a PhD candidate with an entrepreneurial at the BC Cancer Agency's Genome Sciences Centre in Vancouver, Canada. And our lab primarily develops methods and tools for de novo genome sequence assembly. Lately, we've been focusing on large genomes like conifer genomes, which have 20 gigabase genomes in some cases, and looking at assembling 10x genomics chromium data. Today, I'll be talking about a tool I most recently developed called Tigment, which is for correcting misassemblies in de novo assemblies using 10x chromium data, or linked reads in general. So quick background on 10x genomics chromium linked reads. Linked reads are a relatively new data set where you first isolate high molecular weight DNA and then isolate these large molecules in small partitions and barcode those partitions so that when you sequence this data, any two reads that came from the same large molecule will have the same barcode. So one way I describe this is it's like paired end sequencing. With paired end sequencing, you had a molecule of about a kilobase and you got one read from each end. With linked reads, the molecule is much larger, say tens to hundreds of kilobases, and instead of getting just two reads, one from each end, you get about 100 reads, say, distributed evenly across the molecule. So when aligning to a reference genome, typical applications of this new data type is to be able to call variants in repetitive regions, which is difficult with paired end sequencing, to be able to identify structural variants, and also to phase variants across large haplotype blocks. And these are all typically reference-based analyses. This is a relatively new data type, so the software tool space is still evolving and being developed. 10x Genomics themselves have their software suite, which is Long Ranger for reference-based alignment and structural variant calling, phasing, variant, all that fun stuff, and their reference-free assembler Supernova. There's a couple of tools for calling structural variants, Grok SV and Top Sorter, and finally scaffolding reference-free de novo genome sequence assemblies using ARCS, which is a tool developed in our lab, and two other tools, Architect and FragScaf, which were actually developed for other linked read-like protocols, so before 10x chromium data, but can still be applied by tweaking parameters. And there's a preprint available for ARCS. So we've recently started working with this linked read data set in our lab. Two tools that we have are like ARCS, which I mentioned, for scaffolding large genomes, or any size genome, I suppose. And there's a poster for that that my colleague Ben van der Valk is presenting at B48. It's more generally about ABIS 2.0. And also trying to assemble these linked reads data set are sub 1x on any individual molecule, but typically you get about 40% coverage of the molecule. So another application we're looking at is trying to assemble the full sequence of that molecule using a tool that we've already published for a different purpose called Collector. And so we're repurposing that tool for this application. And there, my colleague Justin Chu is presenting that poster on Collector at B40. So my game that I enjoy playing is genome sequence assembly. And when assembling reads into contigs come to an end typically for one of four reasons. Either a large repeat, a gap in your sequencing data due to perhaps GC bias, heterozygous, possibly structural variation, and finally misassemblies. So there's a number of scaffolding algorithms out there. It's an active field of research, and that attempts to scaffold over large repeats and attempts to scaffold over sequencing gaps, but these algorithms typically don't address misassemblies. So if you had the perfect scaffolding tool that does its job ideally and resolves all your large repeats that it's able to, and all your sequencing gaps, then what you're going to be left is misassemblies. So it's these misassemblies at that point that are limiting the contiguity of your assembly, not just the correctness. People typically focus on the correctness of your assembly, but misassemblies also limit the contiguity of your assembly when you have a highly contiguous assembly. So just as a quick cartoon of this, If you have a genome that has 2 linear chromosomes and you've got 1 misassembly, you now got three contigs, and no scaffolding is going to get you from these 3 contigs to your answer, which is the 2 linear chromosomes. So to be able to complete this assembly, to finish it, you're going to have to first correct that misassembly, break that chimeric contig in 2, before your scaffolding algorithm will be able to do its job and put those 2 pieces back together. Well, 4 pieces back together into your 2 linear chromosomes. So this is where Tigment comes in. So it uses linked reads data from 10x genomics Chromium to identify and correct misassemblies by breaking scaffolds at those breakpoints. It does this by counting the number of large molecules that support each position in the assembly, and also looks for molecules that refute the fact that the assembly is correct at that point. And then it outputs positions of those misassemblies. Or putative misassemblies. So the input to Tigment is alignments of your reads to your draft genome, and those alignments can either be done with long ranger, if you like. There are liners called Lariat, or alternatively you can demultiplex the barcodes using long ranger basic, which is a faster tool to run, and use BWA mem to do your alignments. Both are possible. So here's an example of what the data looks like. The top line there is your depth of molecule coverage, so not read coverage, but molecule coverage. And it's reasonably uniform over long distances, much more uniform than read coverage data. And where you have a misassembly, you see these stark drop-offs in molecule coverage. And in addition, at those misassemblies, you expect to see linked reads that are clipped. So that means that the terminal reads of your molecule are like soft clipped alignments. And where you see a bunch of these clipped molecules at one position, it's an indicator that that particular base position is where the misassembly occurs. So with that, you can actually refine your misassembly coordinates down to base pair accuracy. So in this plot, the 3 vertical blue lines are the positions where Tigment has identified misassemblies in this scaffold. One other plot it outputs is this scatter plot of molecule start and molecule end position. So in this, the size of each dot is a molecule, and its vertical distance from the diagonal is the size of the molecule. And some types of misassemblies don't affect your depth of coverage, for example, an inversion. So that would be difficult to detect from just the depth of coverage plot alone. But those types of misassemblies can be identified from the scatter plot, so it's quite useful for that. Another figure output by Tigment is this graph of segments. So here I've taken the scaffold and broken the scaffold into 10 kilobase segments of DNA. So each vertex is a 10 kilobase segment. And that was the input to Tigment. I haven't told it which segments were originally located with each other in the scaffolds. It's just given these 10 kilobase segments. And then it looks for segments that have a large number of barcodes in common, chromium barcodes in common. And if it sees a significant enrichment in barcodes between 2 segments, it joins them with an edge. And then this graph is given to GraphViz to lay out and display. And then here I've colored each vertex with the color of the scaffold that it originally came from. So you can see that Tigment has recapitulated what the original scaffolds were in most of the cases. So the assembly is largely correct. And let's see if this works now. All right, and in positions where you have like a little vertex here that's connected to an otherwise long chain, what that shows is that typically is that you have a scaffold gap in the assembly here, so a gap of ends, and then you have a small contigs that should go in that gap of ends. And you see that actually happens a few times in this assembly, once up there, once down there, and once over here. And you also see, on these 2 little guys over there, and you also see a position that is a potential misassembly, where you have this yellow scaffold and all the coincident barcodes that now join in with this green scaffold. So that's an indicator that this green scaffold might be incorrect in this position, and this 10 kilobase segment should actually be split into 2, one of which might be connected to this yellow scaffold. So there's all sorts of wonderful types of ways that an assembler can go wrong. So my menagerie of misassemblies is chimeric sequence, just joining two things together that shouldn't be together at all. Missing sequence, which is typically a gap in your misassembly, or sometimes if it's a gap of ends, it's correct. If there's no gap of ends, then it's an incorrect deletion or a misassembly. And both of those result in a drop-off in depth of molecule coverage. So these are the types of misassemblies that Tigment can currently identify. Other types of misassemblies are a chimeric insertion, so some other sequence inserted where it shouldn't be, but either side of it's correct, an inversion, and a collapsed tandem repeat. And because these don't result in a change in molecule depth of coverage, each of these is relatively neutral to depth of coverage, and the collapsed repeat actually results in an increase, apparent increase in depth of coverage. Tigment can't currently identify these types of misassemblies because it doesn't. Although it displays that scatter plot, it doesn't use it currently, so that would be future work to be able to identify these types of misassemblies. So this is an example, this is an Igv plot to show some of the types of misassemblies that Tigment can identify. The top plot here is the molecule depth of coverage, and you can see a sharp drop-off where there's the misassembly, and a bunch of molecules starting at exactly that position. So this is the signature for a typical misassembly. And one thing to notice is that although the molecule coverage is quite uniform across a correctly assembled scaffold, and you see that, and the sharp drop-off in coverage is very apparent in the molecule coverage, in the read coverage, you see basically no change. If you're looking at just the reads, the assembly looks correct. So this linked read data really does help highlight where these misassemblies occur that you can't see necessarily from the reads alone. So this misassembly was at the beginning of a scaffold. Here's another one in the middle of a scaffold. Here's another one at the end of a scaffold. And you can see they all display the same signature. So we recently published the Abyss 2.0 paper in Genome Research in the special Genome Assembly issue. And this is a Circos plot of alignments, so scaffold positions versus reference positions. And we like to call these Jupyter plots for, I guess, what's an obvious reason. And these plots, by the way, have been generated by my colleague Justin Chu, so thank you for that. And he did some of these last night, by the way, so I owe him a beer for that. And where you see these lines crossing, that's interchromosomal chimeric sequence. So in what would be a healthy person, probably a misassembly. Now, when we correct this assembly with Tigment, using a minimum of 4 clipped molecules as the threshold to identify misassembly, we remove some of these misassemblies. When we reduce that threshold to 3, we remove even more. And finally, when we reduce that threshold to a minimum of 2 clipped reads at one position, all of the interchromosomal misassemblies have been cleared up. So I think that's an important result. What it shows is that every single interchromosomal misassembly in this assembly was in a low depth of coverage region with at least 2 clipped molecules. So we know this is like at least a minimum. It's a necessary condition for an interchromosomal misassembly in this assembly. Another application we looked at is the Sitka Spruce assembly. We have 10x genomics chromium sequencing of Sitka Spruce, which is a 20 gigabase genome. And we will be assembling the whole nuclear genome using this method in Abyss 2.0. And as a proof of concept and to be able to do library QC, I've been assembling the Sitka Spruce mitochondrion. So the mitochondrial genome of Sitka Spruce is 5 to 6 megabases, so like megabases, so that's huge compared to like human or most animals that has a 16 kilobase mitochondrion. This is the size of an entire bacterium. And using Abyss and ARCs, we assembled it into 16 scaffolds with an N50 of a half megabase. And no matter how I tweaked the ARCs parameters and tried to get a better scaffolded assembly, that is the best I could do. And I didn't think this was as good as we could get, so that's when I started looking into misassemblies as possibly a limiting factor. And so after developing Tigment and correcting this assembly, and scaffolding it with ARCs, we were able to reduce it from to 4 scaffolds, just 4 scaffolds. And 70% of the genome is in that single largest scaffold you see. So the N50 improved 9-fold from half a megabase to 4 megabases. And if you want to see more about this, you can come to my poster at B66, which is, I think, at 1 o'clock. And just as a matter of scale, that little circular config in the bottom right corner is the plastid, so 120 kilobase plastid. So, these genomes are unusually big, and we're not quite sure why yet. So that is the end of my talk, and thank you very much for your attention. Tigment's available online. There's one more slide after this, but there's not. Okay. That's okay. It just had links to these slides and whatnot. So these slides are online at sjackman.ca. And Tigment is online at github.com/bcgsc/Tigment. Thank you. I'll be happy to take questions now.
Thank you. So we have time for a few questions. Please.
Thanks for the talk. Did you try to check the quality of the Supernova assemblies?
Yes, I did.
What did you find out?
I found that the results are generally quite good in terms of correctness. I had a Jupyter plot that I didn't show here, and that displays these interchromosomal misassemblies, and there was only 2 misassemblies. Which is pretty good. Interchromosomal ones, that is. There might be more other non-interchromosomal misassemblies. And Tigment was able to correct one of those 2 with at least the first. I only tried 1 parameter set. With other parameters, maybe more. But yeah, I corrected one of those 2.
Okay, any more questions? Okay, then I suggest to move on. And so we are running out of time a bit.
Thank you.
Okay.